I am Dr. Khokon Kumar Dotto. I have a Christian name, Dr. David Broom. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Roles of Boron in Antioxidant Mechanisms, a story about some unpublished scientific works. My life as a researcher in Indiana University, Bloomington, USA, was unfortunately very short. I worked in the school of optometry as a research associate. Literally, my opportunity in Indiana University was connected with the commitment to find out the mechanisms of cellular death in eye diseases in humans. How pre-radical induced stresses or oxidative stress lead the death of ocular cells, especially the cells of the inner layer of the cornea. At that point, it was known from the work of Dr. Iranga N. Vitana and her group that dysfunction with the genes SLC4A11 or NABC1 are responsible for eye disease named Fuchs endothelial corneal dystrophy. SLC4A11 was known at that point as a Boron transporter. A lot of the functions and other details of SLC4A11 are now known. SLC4A11 is known as multifunctional protein and related with the transportation of different other molecules and ions. Different variants of SLC4A11 have been discovered by the scientists till today. The functional locations and diverse functions of the variants have been explored to some extent. SLC4A11 is known to act as an important factor for other eye diseases also, especially like recessive corneal endothelial dystrophy 2, corneal dystrophy, Harboin syndrome, or perceptive deafness, and others. In plants, bone toxicity is a well-known big issue for scientists for a long time. At present, bone supplement is available in the market, which is recommended for good health for the humans. But at the time in 2009, almost nothing was known to me and my group about the roles of boron and bone-containing compounds in antioxidant mechanisms. In 2020, I could find some research works related with antioxidant properties of boron and bone-containing compounds published before 2008. My experimental approaches were different from others. I tried to check the direct effect of boron and bone-containing compounds in an environment with the presence of high levels of free radicals. Nobody actually tried to verify the effects of boron and bone-containing compounds in that way before. And I found a very complex effects of boron and bone-containing compounds on the survival of corneal endothelial cells, which was largely dependent on the concentration of boron and bone-containing compounds. Boron not only supports the survival of the living cells, it also induces the death at certain concentrations. Nobody tried to look in such a critical and complex way before. I could get help from another professor in the USA with the method to prepare cell growth medium in a bone-free state. Then I tried to look the effect of boron in different concentrations on the survival of the cultured bovine corneal endothelial cells. I found that bones support the growth of cultured bovine corneal endothelial cells only within certain concentrations. Both lower and higher concentration of boron out of that limit act as harmful for living cells and even kill them. But the deficiency of boron and the high presence of boron cause harm to the living cells in a different mechanisms. The death of living cells in a bone deficient environment may be related with the necrotic cell death. The death of the living cells in the presence of high level of boron possibly induce a death related to apoptosis. I tried to induce the high production and accumulation of free radicals in the living cells using different chemicals and approaches, and then try to check the effect of boron and bone-containing compounds in that environment. I found huge neutralizing effects of boron and bone-containing compounds on the oxygen-containing radicals, but I did not find noticeable effect of boron and bone-containing compounds on the nitrogen-containing radicals. I could find a picture with corneal endothelial cells under bone toxicity, which is usually seen in Fuchs corneal endothelial dystrophy. At certain stage, I found dense growth of corneal endothelial cells and cells with more than one nucleus, which is seen in Fuchs corneal endothelial dystrophy. The findings were almost unbelievable something. I tried to reproduce the results of the experiments and succeeded with that. I finally presented the findings to my group in the university, but such a surprising findings never got published by the group after I left the laboratory. Till 2020, many groups around the world have published their exciting works relating the roles of boron in cellular antioxidant mechanisms. Finally, I would like to wish a very happy, prosperous, and dignified life for myself and for you everybody in the light of the true values for freedom. Thank you so much.